Hello everyone, this brand new semester is going to be really exciting. Here we will be learning about biotechnology, some techniques related to biotechnology, and in depth topics like vectors, recombinant DNA technology, like that. So, this particular video is about some introductory topic like fractionation of nucleic acids. Any systematic fractionation method must exploit differences between members of a mixture to effect separation. Nucleic acid molecules can differ from one another in overall size, base composition, topology, and nucleotide sequence. Accordingly, fractionation methods for nucleic acids are based on these features. So, separation of DNA by gel electrophoresis of the various techniques used in the fractionation of proteins. One of them, gel electrophoresis, is also widely used to separate nucleic acids of different molecular mass, nucleotide length. Small RNA or DNA molecules of a few hundred nucleotides or less are generally separated by polyacrylamide gel electrophoresis. Then larger molecules have trouble making their way through the cross-linking polyacrylamide and are generally fractioned on agarose gel, which have greater porosity. Agarose is a polysaccharide extracted from seaweed. It is dissolved in hot buffer, poured into a mold, and caused to gelate simply by lowering the temperature. Separation of DNA molecules greater than about 25 kb is generally accomplished using the technique of pulsed field electrophoresis, in which the direction of the electric field in the gel is periodically changed, which causes the DNA molecules to reorient themselves during their migration. So, following electrophoresis, the DNA fragments in the gel are visualized by soaking the gel in a solution of stain such as etherium bromide. Etherium bromide intercalates into the double helix and causes the DNA bands to appear fluorescent when viewed under ultraviolet light. The sensitivity of gel electrophoresis is so great that DNA or RNA molecules that differ by only a single nucleotide can be separated from one another, a feature that gives rise to an invaluable method for DNA sequencing. Since the rate of migration through a gel can also be affected by the shape of the molecule, electrophoresis can be used to separate molecules with different conformations, which are circular and linear or relaxed and supercoiled forms. So, separation of nucleic acids by ultracentrifugation, when we conduct that, common experience tells us that the stability of a solution or suspension depends on its components. Cream floats to the top of raw milk. A fine particle, a fine precipitate gradually settles to the bottom of a container, and a solution of sodium chloride remains stable indefinitely. Numerous factors determine whether or not a given component will settle through a liquid medium, including the size, shape, and density of the substrate, and the density and viscosity of the medium. If a component in a solution or suspension is denser than its medium, then centrifugal force causes it to become concentrated towards the bottom of a centrifuge tube. Larger particles sediment more rapidly than smaller particles of similar size and density. The tendency for molecules to become concentrated during centrifugation is counteracted by the effects of diffusion, which causes the molecules to become redistributed in a more uniform random arrangement. With the development of ultracentrifuges, it has become possible to generate centrifugal force greater than 500,000 times the force of gravity, which is sufficient to overcome the effects of diffusion and cause macromolecules to sediment towards the bottom of a centrifuge tube. Centrifugation proceeds in a near vacuum to minimize frictional resistance. DNA and RNA molecules have been extensively analyzed by techniques utilizing the ultracentrifuge. For the present purpose, we will consider two centrifugation techniques used in the studies of nucleic acids that have been discussed, like velocity sedimentation. The rate at which a given molecule moves in response to centrifugal force in a centrifuge is known as its sedimentation velocity. Since the sedimentation velocity changes as the centrifugal force changes, a given molecule is characterized by a sedimentation coefficient, which is its sedimentation velocity divided by the force. Throughout this book, we have referred to various macromolecules and their complexes having a particular S value. The unit S, or Swedberg, after the inventor of the ultracentrifuge, is equivalent to a sedimentation coefficient of 10 raised to minus 13 second, because the velocity at which a particle moves through a liquid column depends on a number of factors, including shape, and determination of sedimentation coefficients does not by itself provide the molecular mass. However, as long as one is dealing with the same type of molecule, the S value provides a good measure of relative size. For example, the 3 ribosomal RNAs of E. coli, the 5S, 16S and 23S molecules have lengths of 120, 1600 and 3200 nucleotides respectively. Then equilibrium centrifugation. In the other type of centrifugation technique, equilibrium or isopycnic centrifugation, nucleic acid molecules are separated on the basis of their buoyant density. In this procedure, one generally employs a highly concentrated solution of the salt or heavy metal cesium. The analysis begins by mixing the DNA with the solution of cesium chloride or cesium sulfate in the centrifuge tube and then subjecting the tube to extended centrifugation. During the centrifugation, the heavy cesium ions are slowly driven towards the bottom of the tube, forming a continuous density gradient through the liquid column. After a time, the tendency for cesium ions to be concentrated towards the bottom of the tube is counterbalanced by the opposing tendency for them to become redistributed by diffusion, and the gradient becomes stabilized. So as the cesium gradient is forming, individual DNA molecules are driven downwards or move buoyantly upward in the tube until they reach a position that has a buoyant density equivalent to their own, at which point they are no longer subject to further movement. The next is about recombinant DNA technology, a very important topic. Over the past 30 years, tremendous advances have been made in the analysis of eukaryotic genomes. This progress began as molecular biologists learned to construct recombinant DNA molecules, which are molecules containing DNA sequences derived from more than one source. So recombinant DNA can be used in many ways. We will begin by considering one of the most important applications, the isolation from the genome of a particular segment of DNA that encodes a particular polypeptide. First, however, it is necessary to consider a class of enzymes whose discovery and use has made the formation of recombinant DNA molecules possible. Restriction endonuclease. During the 1970s, it was found that bacteria contained nucleases that would recognize short nucleotide sequences within duplex DNA and cleave the DNA backbone at specific sites on both strands of the duplex. These enzymes are called type 2 restriction endonucleases or simply restriction enzymes. They were given this name because they function in bacteria to destroy viral DNAs that may enter the cell, thereby restricting the growth of the virus. That's why they are called restriction endonuclease. The bacterium protects its own DNA from nucleolytic attack by methylating the bases at susceptible sites, a chemical modifications that block the action of the enzymes. Some sort of epigenetic modification, methylation. So enzymes from several hundred different prokaryotic organisms have been isolated that, however, recognize over 100 different nucleotide sequences. The sequence recognized by most of these enzymes are 4 to 6 nucleotides long and are characterized by a particular type of internal symmetry. Consider the particular sequence recognized by the enzyme EcoR1, 3' CTTAAG 5', 5' GAATTC 3'. The segment of DNA is said to have two-fold rotational symmetry because it can be rotated 180 degrees without change in its base sequence. Thus, if one reads the sequence in the same direction, 3' to 5' or 5' to 3', on either strand, the same order of bases is observed. A sequence with this type of symmetry is called a palindrome. When the enzyme EcoR1 attacks this palindrome, it breaks each strand at the same site in the sequence, which is indicated by the arrows between A and G residues. Red dot indicate the methylated bases. These red dots are actually situated on the A site. So, red dots are on A. This indicates the methylated bases in the sequence that protect the host DNA from enzymatic attack. I mentioned earlier, we need to protect that place from cutting so that we just methylate, not be just bacteria. Some restriction enzymes cleave bonds directly opposite to one another on the two strands, producing blunt ends. Some restriction enzymes cleave bonds directly opposite one another on the two strands, producing blunt ends, whereas others, such as EcoR1, make staggered cuts. The discovery and purification of restriction enzyme have been invaluable in the advances made by molecular biologists in recent years, since a particular sequence of 4 to 6 nucleotides occur quite frequently simply by chance. Any type of DNA is susceptible to fragmentation by these enzymes. The use of restriction enzymes allow the DNA of a human genome or that of any other organism to be dissected into a precisely defined set of specific fragments. Once the DNA from a particular individual is digested with one of these enzymes, the fragments generated can be fractioned on the basis of length by gel electrophoresis. Different enzymes cleave the same preparation of DNA into different sets of fragments, and the sites within the genome that are cleaved by various enzymes can be identified and ordered into restriction maps such as restriction map. So, formation of recombinant DNAs. Recombinant DNAs can be formed in a variety of ways. In the method, DNA molecules from two different sources are treated with a restriction enzyme that makes staggered cuts in a DNA duplex. Staggered cuts leave short single-stranded tails that act as sticky ends that can bind to a complementary single-stranded tail on other DNA molecule to restore a double-stranded molecule. So, one of the DNA fragments that will make up the recombinant molecule is bacterial plasmid. So, bacterial plasmid, some restriction enzymes splits both types of DNA, human chromosome. Then human DNA fragment joins with plasmid by sticky ends. Nicks are sealed by enzyme. This creates a plasmid. So, formation of a recombinant DNA molecule. In this example, a preparation of bacterial plasmid is treated with a restriction enzyme that makes a single cut within each bacterial plasmid. This same restriction enzyme is used to fragment a preparation of human genomic DNA into small fragments because they have been treated with the same restriction enzyme. The cleaved plasmid DNA and the human DNA fragments will have sticky ends. And these two populations are incubated together. The two DNA molecules become non-covalently joined to each other and are then covalently sealed by DNA ligase, forming a recombinant DNA molecule. Okay, so that will make up the recombinant molecule is a bacterial plasmid. So plasmids are small circular double-stranded DNA molecules that are separated from the main bacterial chromosome. The other DNA fragment is obtained from human cell following treatment with the same restriction enzyme used to open the plasmid. And the human DNA fragments and the treated plasmid are incubated together in the presence of DNA ligase. The two types of DNAs become hydrogen bonded to one another by their sticky ends and are then ligated to form circular DNA recombinants. And the first recombinant DNA molecule were formed by this basic method by Paul Berg, Herbert Boyer, Annie Cheng, and Stanley Cohen of Stanford University and the University of California, San Francisco, marking the birth of modern genetic engineering. By following the procedure just described, a large number of different recombinant molecules are produced, each of which contains a bacterial plasmid with a segment of human DNA incorporated into its circular structure. Suppose you are interested in isolating a single gene from a human genome, for example, the gene that codes for insulin. Because your goal is to obtain a purified preparation of one type of recombinant DNA that contains the insulin coding fragment, you must separate this one fragment from all of the others. This is done by a process called DNA cloning. We will return to the search for the insulin gene after describing the basic methodology behind DNA cloning. So we need to learn about DNA cloning. DNA cloning is a technique to produce large amounts of a specific DNA segment. The DNA segment to be cloned is first linked to a vector DNA, which is a vehicle for carrying foreign DNA into a suitable host cell such as the bacterium E. coli. The vector contains sequences that allow it to be replicated within the host cell. Two types of vectors are commonly used to clone DNAs within bacterial hosts. In one approach, the DNA segment to be cloned is introduced into the bacterial cell by joining it to a plasmid as described previously and then causing the bacterial cell to take up the plasmid from the medium. In an alternative approach, the DNA segment is joined to a portion of the genome of the bacterial virus lambda, which is then allowed to infect a culture of bacterial cells, producing large number of viral progeny, each of which contains the foreign DNA segment. Either way, once the DNA segment is inside a bacterium, it is replicated along with the bacterial or viral DNA and partitioned to the daughter cells or progeny viral particles. Thus, the number of recombinant DNA molecules increase in proportion to the number of bacterial cells or viral progeny that are formed. From a single recombinant plasmid or viral genome inside a single bacterial cell, millions of copies of DNA can be formed within a short period of time. So once the amount of DNA has been sufficiently amplified, the recombinant DNA can be purified and used in other procedures. In addition to providing a means of amplifying the amount of particular DNA sequence, cloning can also be used as a technique to isolate a pure form of any specific DNA fragment among a large heterogeneous population of DNA molecules. We will begin by discussing DNA cloning using bacterial plasmids. So cloning eukaryotic DNAs in bacterial plasmids. This foreign DNA to be cloned is first inserted into a plasmid to form a recombinant DNA molecule. Plasmids used for DNA cloning are modified versions of those found in bacterial cells. Like the natural counterparts from which they are derived, these plasmids contain an origin of replication and one or more genes that make the recipient cell resistant to one or more antibiotics. So, antibiotic resistance allows investigators to select for those cells that contain the recombinant plasmid. So, as first demonstrated by Avery, MacLeod, and McCarthy, bacterial cells can take up DNA from their medium. This phenomenon forms the basis for cloning plasmids in bacterial cells. In the most commonly employed technique, recombinant plasmids are added to a bacterial culture that has been pre-treated with the calcium ions. When subjected to a brief heat shock, such bacteria are stimulated to take up DNA from their surrounding medium. Usually only a small percentage of cells are competent to pick up and retain one of the recombinant plasmid molecules. Once it is taken up, the plasmid replicates autonomously within the recipient and is passed on to the progeny during cell division. So those bacteria containing a recombinant plasmid can be selected from others by growing the cells in the presence of the antibiotic to which resistance is conferred by one or more genes on the plasmid. So we begin this discussion with the goal of isolating a small DNA fragment that contains the sequence for the insulin gene. So to this point we have formed a population of bacteria containing many different recombinant plasmids, very few of which contain the gene being sought. So one of the great benefits of DNA cloning is that in addition to producing large quantities of particular DNAs, it allows one to separate different DNAs from a mixture. It was noted above that plasmid containing bacteria can be selected from those without plasmids by treatment with antibiotics. Once this has been done, the plasmid bearing cells can be grown at low density on petri dishes so that all of the progeny of each cell, a clone of cells, remain physically separate from the progeny of other cells. Because a large number of different recombinant plasmids were present initially in the medium, the different cells plated on the dish contain different foreign DNA fragments. Once the cells containing the various plasmids have grown into separate colonies, The investigator can search through the colonies for the few that contain genes being sought. In this case, the insulin gene. So, culture dishes that contain bacterial colonies or phage plaques are screened for the presence of a particular DNA sequence using the combined technique of replica plating and in situ hybridization. So, replica plating allows one to prepare numerous dishes containing representatives of the same bacterial colonies in precisely the same position on each dish. One of the replica plates is then used to locate the DNA sequence being sought, a procedure that requires that the cells are lysed. So we purify human DNA. E. coli chromosome is there, plasmid is there, antibiotic resistance gene is there in that E. coli chromosome, and purify plasmid DNA. Then treat with EcoR1 to cleave both human and bacterial DNA into fragments. Then join that fragments into recombinant DNA with DNA ligase. So, population of plasmids containing different segments of human DNA incubate E. coli cells under conditions in which they will take up plasmids from the medium, grow cells in medium that selects for those containing a recombinant plasmid, insulin gene, ribosomal RNA gene. So, this is an example of a DNA cloning using bacterial plasmids. DNA is extracted from human cells, fragmented with EcoRI, and the fragments are inserted into a population of bacterial plasmids. Techniques are available to prevent the formation of plasmids that lack a foreign DNA insert. Once a bacterial cell has picked up recombinant plasmid from the medium, the cell gives rise to a colony of cells containing the recombinant DNA molecule. In this example, most of the bacteria contain eukaryotic DNAs of unknown function, while one contains a portion of DNA encoding ribosomal DNA, RNA, and the other contain DNA encoding insulin. So later when we learn that separation of plasmid DNA from that of main bacterial chromosome by CSCI, cesium chloride equilibrium centrifugation. This centrifugation tube have been seen to contain two bands, one of plasmid DNA carrying a foreign DNA segment that has been cloned within the bacteria, and the other containing chromosomal DNA from the same bacteria. The two types of DNA have been separated from centrifugation. The researcher is removing the DNA from the tube with a needle and syringe. The DNA in the tube is made visible using the DNA binding compounds ethidium bromide, which fluorescent under ultraviolet light. So, how to create, located a bacterial colony containing a desired DNA sequence by replica plating and in situ hybridization. Sterile filter paper or velvet dish, culture dish with bacterial colonies of phage plaques containing recombinant DNA bacterial colony. Transfer representative cells to nitrocellulose membrane by replica plating technique. Then lyse cells and treat to denatured DNA and cause single stranded DNA to adhere to the filter in place. Denatured DNA adhering to membrane incubate the radioactivity or fluorescently labeled probe position of labeled hybrid. So bacterial cells that were plated on the culture dish have grown into colonies. The dish is inverted over a piece of filter paper, allowing some of the cells from each colony to become absorbed to the paper. Empty culture dishes and then inoculated by pressing them against the filter paper to produce replica plates. So procedure for screening the cells of a culture dish for those colonies that contain the recombinant DNA of interest. Cultures can be screened using either radioactively or fluorescently labeled probes. Once the relevant colonies are identified, cells can be removed from the original dish and grown separately to yield large quantities of DNA fragments being sought. Then fixed onto a surface of a nylon or nitrocellulose membrane. Once the DNA is fixed in place, the DNA is denatured to prepare it for in situ hybridization, during which the membrane is incubated with a labeled single stranded DNA probe containing the complementary sequence to that being sought. So following incubation, the unhybridized probe is washed from the membrane and the location of the labeled hybrids is determined. Live representation of the identified clones can be found at corresponding sites on the original plate, and these cells can be grown into large colonies which serve to amplify the recombinant DNA plasmid. Then after the desired amount of amplification, the cells are harvested, the DNA is extracted, and the recombinant plasmid DNA is readily separated from the much larger chromosome by various techniques including equilibrium centrifugation, which we mentioned. The isolated recombinant plasmids can then be treated with the same restriction enzyme used in their formation, which release the cloned DNA segments from the remainder of DNA that serve as a vector. The cloned DNA can then be separated from the plasmid by centrifugation.